Remember, the content on our show is for information only.
It is not intended to provide medical or other professional advice. Hi, and welcome to Placebo, the podcast where we take turns teaching each other something new or interesting about healthcare or medicine or sciencey type things related to that.
Yeah, whatever.
I'm Kaylin Richards, and this is my brother.
And I'm Logan Richards.
Hello, brother.
Hello.
And today we have a very specific topic that I've researched because I'm trying not to make super long episodes.
Okay, good.
Especially because my puppy's waiting to go out and run right now.
Mm-hmm.
So, you remember we've talked about telomeres before, or telomeres. I'm not really sure what the right pronunciation is.
I've heard it both ways.
We should agree on one because I'm going to say it a lot right in this episode.
Okay, hold on.
Okay.
Telomere.
Telomere.
Telomere. There's an O in it.
But she said telomere.
Telomere.
See? She said telomere. We've talked about them before.
Okay.
I think it was from the episode about aging, maybe?
Sure, yeah, that sounds right.
Okay, do you remember anything about them?
They're the end of the chromosome.
They're the end of the chromosome, yes. So, right, in all of our cells we have chromosomes. How many do we have?
We have 23 pairs.
23 pairs. So in most humans, in most cells, there'll be 46 chromosomes.
Yes.
But there will be exceptions to that, of course. So these contain the DNA, right? That we need to
That's what I've heard.
Function. Yes, that's what I've heard. I haven't seen it for myself, but that's what I've been told. And so
you never did the experiment in biology class where you got to spin up your own DNA, you know?
Yeah, but it just looked like strings. It didn't look like chromosomes.
Yeah, but that's the DNA.
Yeah, okay. Did do that. So back on focus. So we have the chromosomes. They have all of our DNA that our body needs to function and survive and live.
Mm-hmm.
And on the end of each chromosome is the telomere. Is that how we decided to pronounce it?
Or telomere.
Telomere. Okay.
Maybe there's an American and a European pronunciation.
I don't know. So it's kind of like a chunk of DNA that maybe doesn't do much of anything. And I'm not going to get too much into the maybe in this episode. That's a whole other thing. Maybe it does some things, but for the most part, probably doesn't do too much. Except it's kind of a placeholder at the end of the chromosome. And that each time a cell divides, and therefore each chromosome divides, a little bit of it is broken off and is gone.
Okay.
Okay, so over the lifetime of the cell, and therefore like you, the person...
How long does each cell live?
It depends on the type of cell, right? So a skin cell has a much shorter lifespan than a neuron.
Well, see, you've brought up the first point that I already don't understand. So it's my understanding that the DNA will split in half, right? Mm-hmm. Using the RNA polymerase or whatever.
Oh, let's not get too fancy here.
It will split in half.
Okay.
And half of the DNA will go to one of the new cells, and half will go to the other.
But it's duplicated.
Yeah, well, so on the side, there'll be a new one.
Yes.
So am I to understand that there is at least one, and probably two, right, pieces of DNA that are from the originals? Does that make sense?
No. What are you talking about?
Well, because each time it's copied, mm-hmm. there's two new pairs, there's two new chromosomes, whatever.
Okay.
Whatever you want to call them. And half of that chromosome is the actual part of the pairing from the original one.
Yeah.
So, does that mean that chromosomes never go away since they're always splitting and then moving on to being part of a new cell? But are they the original chromosomes?
Well, it's diluted down. Some part of it is, but
I don't think I've ever asked this question.
So here you have one cell, and it's got an A side and a B side.
Yeah.
And the DNA, unzips.
Right.
And then you get a side that's like A, a new A1.
Right.
And then there's the original B and like B1 over here.
Right.
And then when A and A1 divide, there's A and another new part to it, and A1 and A1.
But if we're just talking about like one single strand of chromosome that
mm-hmm.
splits down the middle and then moves over to a new cell, that half strand of chromosome...
It's not a half. It's got the whole other side joins onto it.
I know, but the half of it...
I think we're having a discussion that is not relevant at all to the topic. But okay.
Well, you say the lifespan of the cell, and I want to know what the lifespan of a strand of DNA is. Because if we're talking about telomeres, and telomeres somehow result in aging, it sounds like telomere or DNA, and therefore telomeres, last much longer than an actual cell does.
Yes and no, because it will go into the new cell if it divides.
Half
but
of it will.
Half of it. But if a cell instead destroys itself, apoptosis, when a cell just gets to a point where it's either programmed to destroy itself or it gets, well, we'll get to more of this, but then the whole cell just in various ways dissolves and is no longer a cell. So it could do that.
Okay. What's the answer to my question, though? Yes.
So yes and no.
DNA lasts much longer than the cell itself.
I wouldn't say
assuming
much
that
longer
the cell doesn't
necessarily.
kill itself.
Yeah.
It just seems to me that it has to last longer, right? If these telomere length has something to do with your overall lifespan.
It does last longer.
Then they have to last your lifespan, right? Or at least a significant
Not
portion of
necessarily.
it. Not like a cell, which, what do they say? You have a new whole set of skin cells every 7 years.
Yeah, but don't quote me on this, but I'm pretty sure neurons are not replaced. I'm pretty sure they are there.
Well, I think there's very limited
neuron growth.
Neurogenesis, but there is neurogenesis.
Anyways, this is not the topic at all, so I want to move to what the actual topic is. You kind of took us on I was trying to make a short and snappy episode.
Okay, we'll move on. We'll just take
it's
that
okay.
whole part out.
No, don't. Just leave it in.
Okay.
Leave it in. Okay, so now I'm just on the second point on my notes. Gonna go back to that. So as we talked about with aging, as you age, in general, when the cell replicates and divides, the telomere will be shortened gradually over time.
Okay.
Okay, that's all that we care about right here.
But see, that implies that the DNA strands are lasting longer than the cells. Because there'd be no reason for them to get shorter if they were just going to make a new one.
Okay, well, the chromosome itself, some of the base
This
pairs.
is a question I've never answered, and you've just brought it up.
One half of...
Yeah, this
when
is what I'm saying.
a chromosome is all compacted, right? It's all curled up strands of DNA.
Yeah.
So when it's ready to replicate, this is pulling back to my biology undergrad major here. When it's ready to replicate, it will open out, unspool, and then unzip. So you have each half of the base pair of the DNA double helix.
You got half strands
separated, and then
they build new strands.
the new base pairs will come and join in on each part of the unzipped one until it's completely replicated.
Right.
And then it will separate, itself into two separate chromosomes that will then refold themselves up and be into two new cells.
And that's exactly what I'm saying.
So one part,
exactly half of the DNA is
not exactly. No, because some of the telomere gets ripped off, and that's my point. That's all that matters in this.
But a certain portion of the DNA is from the cell before.
Yes, but
and
if you're talking
how many cells before?
From the time that you're
the cell before and the cell before and the cell before.
It's only one little. Like, if you go out many generations, it's only going to be one tiny fragment. It's very diluted. That's going to be from the cell, 4 generations ago, right?
I don't know. That's why I'm asking.
Yes, that's what I'm saying.
Okay.
Okay. Anyways, let's focus on what the episode is about, which is telomere shortening and how it and why that happens. And there's some weird, kind of trippy things about it. That's what I want to talk about. That's the interesting thing here.
Tell me more.
Okay.
I've given up on understanding this.
That's not what I researched.
DNA thing.
Okay.
Doesn't make any sense.
It does. What doesn't make sense? Anyways.
Okay, telomeres. Telomeres.
I think the Google lady said telomere.
Telomere.
So, as you age, as your cells replicate over time, whatever cells they may be, and however long the same DNA is in it, whatever.
Okay.
The telomere gets shorter and shorter, okay? Until eventually you get past the telomere, and now you delete information that's important.
Oh, that sounds bad.
Yes, it is. So now, when the cell replicates, you're losing more and more information that is, necessary for the cell to survive.
Mm-hmm.
Okay.
Yes.
So first, before we get into more of the consequences about that, I want you to just tell me, why do you think that happens? Not all organisms have a telomere that shortens. Some organisms don't. And some organisms have an enzyme that just rebuilds the telomere after each replication.
Well, that seems incredibly helpful.
So why do you think that we have that?
You're asking me to come up with some type of evolutionary explanation?
Yeah, why do you think it... Yeah, well, because it is of benefit. There is a benefit to it that clearly won out evolutionarily for humans. Why do you think?
It makes sure that we never have too old of DNA strands.
You're really focused on that, and that's not what it is.
It makes sure that it leaves room for mutations, which can be beneficial.
Mutations
sometimes.
is sort of on point, but not entirely. So
Okay.
what I would say is, according to my research, a lot of organisms that have shorter lifespans are the ones that have the telomeres that don't shorten.
Oh.
And organisms like us with longer lifespans have telomeres that shortened more frequently. And the reason, at least one of the reasons that I saw speculated, is to act as a tumor suppressant, so to prevent against cancer, which can be caused by mutations. So when a cell replicates really rapidly, like in cancer, the hope is that if you have a telomere that just shortens pretty quickly with the cell that's replicating really rapidly, that then you would get to the point where the cell would kind of implode and kill itself, and maybe you wouldn't have as many tumors or cancers.
Oh, okay.
That's
Seems like a reasonable
That's the
possibility.
theory. Of course, there's a really significant trade-off, which is aging.
Yes.
Yes. Okay. So, yeah, basically, it's a safety mechanism against cancer. The question is it? I think some people,
Is that the
maybe Aubrey de Grey would argue, is it worth it?
Yeah.
That's not...
Is that the only hypothesis?
That's the only
Or-
one that I saw. I'm not going to say it's the only one. That's the main one that I saw presented.
Okay.
I thought, I like this phrase, that when the telomere gets too short and they're deleting the important information, they call it a genome catastrophe.
Ooh, that does.
It's very dramatic.
That's
Yeah.
It would be, I assume.
So something that I didn't know was that the telomere does not shorten at the same rate for everybody. And there are lots of things in your life that can cause it to shorten more quickly.
Like environmental factors?
Totally.
Oh.
So maybe sometimes I read someone's chart and I notice that they are 50, say, and I go and meet them and I would think, oh, wow, they look like they are 75. I would have thought they were 75 if I didn't know that. Some people age more rapidly, probably based on a lot of factors in their life.
Yes.
And sometimes it's the opposite. Someone looks extremely youthful when I would, if I didn't know their age, I would have guessed they were a lot younger than I thought they were.
Sure.
People age at different rates, and I think a large part of the aging can be related to, or can be called back to telomeres, and a large part of that is environmental factors in your life. So we're going to talk about some of those.
Okay.
Just to kind of give a picture. First of all, I'm going to talk about there are some like genetic conditions, like premature aging conditions.
I've never heard of those.
This one, it lists several: Werner syndrome, Hutchinson-Gilford syndrome, dyskeratosis congenita. I've heard of Werner syndrome. I believe that's the one where you age very rapidly, and by the time you're like 10 or 12, you look like an elderly man or whatever. So it's a really sad condition.
Sounds
they're
that
like
way.
a child in their mind, but an old person in their body.
Yeah.
So all of those conditions lead to accelerated aging and a reduced lifespan because of that. And all of those conditions, people have shorter telomeres from a genetic, mutation, malfunction.
Okay.
So some of the studies looking at like environmental factors and how they affect the telomere length. The first one I'm going to mention is looking at early childhood deprivation. So this looked at kids that were raised in Romania, Bucharest, in like institutional facilities. So,
Okay.
So they didn't get a lot of one-on-one interaction. They didn't get a lot of enriching, experiences.
Sure.
This study found that kids that were raised in this institutional environment up to age 8 had like much shorter telomere lengths than kids that were raised in it after that age, or kids that were never in it.
Well,
So it was like that. There was like an early stage in life where they were most sensitive to getting the shorter telomeres.
this is like the real life version of when something says that'll take 10 years off your life.
Yeah, I don't know how specific we can be about measuring that, but
No. Sounds like it, though.
There's also been some studies looking at neighborhood level social and environmental risk. So like, is your neighborhood dangerous, I guess?
That seems like
Or
it.
polluted or things like that. And also prenatal tobacco exposure. Both of those were leading to shortened telomeres. And both of those are, I think, more focused on early life.
All these topics are so interesting because they're the types of things that you hear economic studies about or public health studies about, or like you hear all those types of studies of oh, there's this type of, environmental factor when you're young, and then this will happen when you're older. But I've never thought of it in
the genetic
very
sense.
specific genetic sense that it will actually could shorten your telomeres.
I hadn't either, because I would think like, oh, maybe you didn't have a supportive environment growing up. Maybe you didn't develop good coping skills because you didn't learn them when you were young. So you have trouble, coping with things later in life, and that you don't do as well as stress. And so that shortens your life maybe, or makes you less successful.
Or I think of like the food desert things where they're saying, oh, you can't get to, fresh fruits and vegetables, and that caused a certain type of malnutrition, and then that affected, so on and so forth. But
maybe what it really is at the base level is affecting your telomeres.
Right.
I should say, I'm not sure why. None of the studies talked about why. So
stress.
Yeah. So they did find that, like, obviously poor caregiving environments for the child, like I talked about in the institutionalized children. Mm-hmm. But they found conversely that if maybe there's like not a great environment, like there is maybe the prenatal smoke, for example, but they had high quality parental care in their early years, that would kind of buffer or negate the effect on the telomeres.
Oh, interesting. Interesting.
Also, there's effects in adults. So adults and adolescents, it's not just kids that can have their telomeres shortened, although there is that kind of more sensitive time when you're really young.
Okay.
But adults or adolescents with cardiovascular disease, with diabetes, with obesity, they all have been found to have shortened telomere lengths compared to other populations that are, normed otherwise.
That just proves it. It's stress.
Maybe. So I guess maybe I should state something that's kind of obvious, but a decreased telomere length has been found to be correlated with poor health outcomes. And
it certainly seems that way.
Yeah. It also may foreshadow health risks, like in the kids that have a shorter telomere, it has like pointed to you probably will have more health issues later on in life.
It doesn't sound like it can be a very...
Because like these young kids, they might not have any health issues themselves, but it might just come out later on in life.
It doesn't sound like it can be very specific, though.
Not necessarily. And then conversely, the other thing is true. Longer telomere lengths have been shown to be correlated with greater improved health outcomes and more longer life and stuff like that. So there's also, there was studies looking at pesticide use in like people where that was their job. They were applying pesticide regularly. And the people that
doesn't sound like a good job.
No. I mean, you would think that they should be wearing protective equipment as appropriate. Maybe they are, maybe they aren't.
Can do, though.
Right, because the people that are doing that regularly did have shorter telomeres, again, compared to a normed other group. And then adults with like sleep apnea, again, shorter telomeres compared to those that didn't. Okay, so...
Can I ask how you measure the length of a telomere?
They took, most of them, they would say they would take, I think you say buccal or buccal sample.
Buccal, right?
Yeah.
No, it's buccal. That's what the dentists say.
From the cheek, a cheek
buccal.
swab. And so they're taking just...
That means the outside of the tooth, the buccal side.
Okay. They're just taking a swab, like like you do from your cheek. Like I got registered with the bone marrow registry. They send you a little cheek swab that you do.
Right.
And you get skin cells from inside your cheek, and they just do a DNA analysis of those.
But beyond that, so are they using microscopes to look at the length, and then they're putting some type of microscopic ruler next to it?
Well, I don't think you can just use a regular microscope and look at a chromosome and measure the telomere. I think you need maybe an electron microscope. I don't know.
I don't have a degree in biology.
I think they measure base pairs, because I did see one article that was listing about like how many base pairs are lost in each cell replication. So I think they can measure at that level.
Because we know where like the stop combination is, right? Isn't there
There is. That's like, this is the end of the telomere, and now we're into the normal.
Yeah.
Okay,
interesting.
So I want to mention one more interesting study. So this was a study with veterans.
Okay.
U.S. military veterans, and they did a study looking at a lot of different health data, but this particular thing that looked at the telomeres was related to hostility. So they found that veterans that had higher scores on a measure assessing for hostility had more shortened telomeres. And then they tried to look more at that because they're like, well, that's kind of random. What's going on there?
What's their measure of hostility?
Oh, I didn't write down what it specifically was. I think it was survey questions.
Okay.
So they did more analysis of that and tried to dig deeper into that. And they found that these were people that specifically had trouble controlling anger, not that they had like aggressive urges or things like that.
Oh, okay.
So they think that this difficulty in controlling anger is associated with the telomere shortening in veterans, and that then prevention, like treatment programs for veterans to like work on controlling anger, would be helpful in preventing telomere shortening in them, and therefore in better health outcomes down the line for veterans.
So very important.
Yeah, so they're thinking like there are programs that do that work on, appropriate control of anger and stuff like that, because I think that can be a difficult thing for the veteran population.
So now they need to figure out, they need to do another study to measure
does it work?
Yeah.
Yeah, and so maybe if veterans that do participate in programs like that, do they have longer telomeres compared to their peers that don't?
That would be quite the advertisement. Live longer.
Live longer, be happier.
Yeah.
So I think we've talked kind of a lot about the telomere shortening and basically how...
Ooh. Are you going to tell me how I can lengthen my telomeres?
Well, here's the thing. Okay.
That's what I'm interested in.
So that's what I was at first looking into. But I started to find that maybe that's not a good thing because, like we talked about to start with, when you have a longer telomere, or in the organisms that don't shorten their telomere, there are other health risks. For example, greater risk of cancer.
Right.
So that is maybe not a good thing and not what we should try and do.
Well, maybe I just want an appropriately lengthened telomere.
So there were some studies looking at that, like maybe there's an enzyme called telomerase, which in some organisms is present and rebuilds the telomere each time.
I like how enzymes are always named appropriately.
ACE,
At least
telomerase.
most of the time.
They're so easy. So there were some studies looking at organisms and how could you use that to just rebuild the right amount and not add too much on, because then you don't want a really long telomere.
That's my goal.
So they're looking at that. Unfortunately, as we've talked about in the past, some of the animal models that are used are not helpful because their system of dealing with telomeres and telomerase is so different to humans. So like mice, for example, use the telomerase, I believe, to keep really long telomeres. Mice have short lifespans, so they don't really work as a model.
Not a very good comparison.
No. There was one article I saw that suggested using a type of fish that has a more similar model to humans. But
interesting.
Anyways, there were a lot of studies with like yeast cells, which are interesting, but not really applicable to people right away, at least. I do want to add one study, though, that is positive, that talks about how you can maybe not shorten your telomeres.
Reduce the...
Shortening.
Reduce the speed of decline.
Yes. Okay. So, like I mentioned, maybe with the very first study, that there's kind of this early stage in early life where you are most sensitive to the shortening.
Do they know when that is, or just early life?
Well, one study looked at before 8 years, said before 8 years,
Right.
and one study said before 4 years. So I would say
sometime up there.
Between 8 and 4.
Okay.
Okay, between birth and like 8 to 4.
4 to 8.
Yeah. So this study looked at the difference in telomere length between babies that had been breastfed and those that had been not.
Okay.
Babies that were breastfed showed longer telomeres compared to their peers in all ways besides they were fed on formula. So breastfeed your baby if you can, and you'll set them up for a better start in life, I guess.
Seems that way.
And with the breastfeeding, it's interesting. There was a specific, very small time window where there was the best effect. So if they only breastfed between the 4th and 6th week of life, so if they only breastfed between like the 4th and 6th week of life, like that 2-week window, then they had like long-term effects with the longer telomere length.
Wow.
So if for some reason you're having trouble, if you can at least breastfeed for those 2 weeks, that's going to have the best long-term effect.
That seems like a quite random time period.
I know, but in early life with, like as an occupational therapist, we learn all the different stages of development, in early life. I bet you learned a lot of those too.
Sure.
There's a lot of very specific windows for different things.
There are, that's true.
So It doesn't surprise me that much. Anyways, that's my episode today. I tried to keep it focused and brief.
That was a good one, though. I think there's so many interesting avenues that you could take, too, because it sounds like this telomere length has to do a lot with population health, like public health, and that we could be doing a lot of things in our public health planning And I wonder if we aimed as our goal to help keep people's telomeres longer, then we would just kind of always end up with better health outcomes, which it sounds like we might.
I think
so.
that's what it, yes. In general, longer telomeres equals better health up to a certain point. If you have a really long telomere, then you're at risk for cancer. So I don't know where the cutoff there is, though.
Yeah.
So,
but it sounds like setting up the right environment. But I wonder if that could lead to some type of innovative public health initiative where they found something that helps keep your telomeres,
at a good length.
The decreasing, stops the decrease a little bit.
Yeah. I guess what you don't want is the rapid decrease associated with some negative environmental.
Some type of stress.
Yeah. Stressors or toxins or whatever, like the pesticides.
Right.
Yeah.
Very interesting stuff, though.
All right.
I very much appreciated the knowledge.
Good. So that was my episode, and we're just at 30 minutes, so Ivy will be so happy to go out to the park now.
Perfect.
If you enjoyed the episode, I hope you did, please consider sharing us with a friend or giving us a super quick little rating on iTunes. Helpful to help other people find the show.
You can visit our website, placebopodcast.com, for show notes or to send us any suggestions or feedback.
You can also find us on Facebook, where we try to have it post every week when the episode has gone up. We're having trouble with that sometimes.
Thank you to James Aiken for our music.
And thank you to you for listening. We'll be back with a new episode next week.
Goodbye.
Goodbye.